To begin a DNA extraction, you'll want to collect some tissue samples. In this example, I will be using leaf tissue collected from common bean plants. I start by labeling some microcentrifuge tubes. This is so that I can keep track of which sample is in each tube. You will then want to collect between 0.05 and 0.1 grams of tissue when working with leaf tissue. It's important to not collect more than this, and I find that approximately a dime-sized amount of leaf tissue is sufficient. After collecting the tissue, I either put the tube rack on ice or I return to the lab immediately. Back at the lab, I review my protocol to make sure that I'm prepared to do all of the steps. I will also write down which samples I will be running an extraction on that day. Next, I will preheat the incubation bath and typically preheat my C-tab buffer as well. With my preheated C-tab buffer, I am ready to begin the first steps of the DNA extraction. I arrange my microcentrifuge tubes with the samples in front of me, along with my C-tab buffer and my pipette. In this step, I will add approximately 250 microliters of C-tab buffer to my microcentrifuge tube. It is important to make sure that I hold the pipette tip upright throughout this process. Otherwise, I might not be getting the appropriate amount of liquid in the pipette tip. If at any point the pipette tip touches the inside of the microcentrifuge tube, I'll need to exchange it for a new one to avoid any contamination between samples. Next, I will use autoclaved pestles to crush the leaf tissue that is now submerged in C-tab buffer. Following the crushing step, you will be able to see the macerated leaf tissue within the tube. After this, you can add another 250 microliters of C-tab buffer. Do not do this second addition of C-tab until after the pestle step to avoid spilling. The samples can then be added to the incubation bath where they will sit for 60 minutes to allow for the further breakdown of tissue. Next, while working within the fume hood, you will add 500 microliters of chloroform to extract the DNA from your macerated and incubated leaf tissue samples. The actual extraction process will take place during this step, and you will shake the tubes with the chloroform and C-tab buffer vigorously for approximately 2 minutes. It is the pestle crushing stage and the chloroform extraction shaking that allows for the highest quantity of DNA in your end product. Because of this, I make sure to time myself so I don't cut any corners and reduce the quality of my ultimate DNA sample. Typically, I also add a paper towel between two tube racks for the shaking in case any sample is to spill out of the tube while it is being shaken. After the 2 minutes of shaking, you will centrifuge your tubes for 15 minutes to separate out an aqueous layer for future extraction steps. It is important to remember to keep your tubes balanced within the centrifuge chamber. An imbalance of the tubes that are not equally spaced can cause issues for the machine over time. For this step and my protocol today, I will be running the centrifuge at 10,000 times G for 15 minutes. While the centrifuge runs, I label new tubes as I will be removing the aqueous layer and putting it into a fresh tube. I will now be removing 300 microliters of the upper aqueous liquid, which you can see above the pelleted leaf tissue here. I bring my freshly labeled tubes directly below my existing samples. This helps me to keep things organized while I am transferring the aqueous layer. Additionally, I only keep one pair of tubes, the old tube and the fresh tube into which the aqueous layer will be put into, open at a time. This is a double step to help me from making any mistakes in the process. During this step, you will want to replace your pipette tips between each sample, as the liquid that you are pulling out from the original sample tube does have unique leaf tissue in each microcentrifuge tube. Again, keeping the pipette tip upright will make sure you are getting the most accurate samples drawn from each tube. After the aqueous layer has been transferred to a new tube, I will precipitate the DNA by adding 200 microliters of isopropanol. If I was unable to get a full 300 microliters of the aqueous layer originally, I will calculate whatever 2/3 volume would be equivalent to, and that is the amount of isopropanol that should be added to precipitate my sample. Since I am using the same tube of isopropanol to add to my microcentrifuge tubes, I'm able to use the same pipette as long as I am cautious not to touch the inside of my sample tubes. Once this step has been done, I invert each tube a few times to mix. After the mixing, I will run my samples in the centrifuge chamber again. As you can see, there's been a separation of aqueous layers, and my sample has likely precipitated to the bottom. I may or may not be able to see a small block of DNA sample at the bottom of my tube at this point. Do not be concerned if you don't see this, as it does not always appear. Next, I will pour off the supernatant, or liquid, on top of my precipitated DNA. And if I am unable to get all of the supernatant removed, I will remove the residual liquid by aspirating or with a pipette. Here you can see there's still a small amount of liquid, and so I will remove it with my pipette set to about 50 microliters. I will repeat the supernatant removal with all of my samples. Following this, I will be adding 300 microliters of 76% ethanol, 10% ammonium acetate solution. Ammonium acetate is used in a DNA extraction to remove impurities and concentrate the DNA that we precipitated in the last step. It is common when working with a pre-made kit or doing a DNA extraction with raw chemicals that you will have wash steps towards the end of the extraction. These steps are intended to remove any proteins or other impurities that you do not want in your DNA. If you do not remove impurities, you may see strange numbers on your 260-280 graphs when you look at your DNA quality following the extraction. After adding ammonium acetate, I will centrifuge my microtubes once again for 15 minutes at 10,000 times G. Following this centrifuge step, I will have a clear supernatant that I will pour off as I did before. Similar to the last time, I will need to remove any residual supernatant by aspirating with a pipette or leaving the samples out for 15 minutes to air dry. Once the tubes are dry, I can add up to 250 microliters of 1 times TE buffer. If you would like to remove your RNA from your DNA sample, now would be the time. Otherwise, you may store your DNA samples in the buffer overnight at 4 degrees Celsius or long-term at -20 degrees Celsius. That's it. Nice job.